Well, this is like 401. Do you want to go ahead
Sure,
and go?
absolutely, yeah. I guess I have to sort of talk a little bit over here. There's the microphone right there. Well, it's my pleasure to introduce Professor Mike Merchant from the Department of Medicine. Mike did his PhD in chemistry at the University of Arkansas and arrived here at UofL in 2003. He's currently the core director for the CIEHS P30 Omics and Exposure Facility Core. He's co-director of the Omics Core for the University of Louisville Alcohol Research Center on Craig McLean's P50 grant. And he's co-director of the Core and Clinical Proteomics Laboratory for the Department of Medicine. He's co-director with me for the NIDDK T35 Summer Research Training Program for medical students. And he has mentored a number of medical students, postdoctoral fellows, graduate, and even undergraduate students. He served on a variety of NIH and DoD and American Heart Association grant review panels. His publication rate is in some sort of logarithmic scale now. He has 124 peer-reviewed research publications and 10 patents. He is the review editor for the journal Renal and Epithelial Physiology. He's currently two MPI on two NIH R01s and a co-investigator on at least 7 other NIH grants. So he's very busy, and appreciate your giving a talk today and telling us about your latest research.
Well, thank you for the invitation. I want to share with you some research that is mainly being conducted by medical students coming through the T35 training grant. Previously, I had presented some work done by Diamond Shelton, who's an MS3 now, looking at the regulation of gene expression and some Epi Transcriptomic marks on histones as a function of confluency for renal epithelial cells and high glucose, the impact of high glucose. Today I wanted to talk about diabetes, epigenetics, and kidney disease, in fact, it's not going to be epigenetics, it's going to be on Epi Transcriptomics. This is a really new area that's evolved, and Dr. Kling is certainly highly accomplished in this area, and Dr. Zhang at Cream Center has developed a mass spectrometry-based methodology for surveilling and quantifying epitranscriptomic marks on nucleosides. And so I want to share with you and relate to you some information that we're developing for diabetic kidney disease. And this work, the majority of the experimental work will be a result of the efforts of Zohaib Haq in MS2 that he completed over this summer. So some of the objectives that I want to review today or address today is to first recap diabetic kidney disease and the idea of metabolic memory. Then very briefly address kind of a working understanding of epigenetics as it relates to modifications to DNA, RNA, and proteins, and then transition from kind of epigenetics to epitranscriptomics and the RNA-OME in diabetes, and lastly, for about half the talk, address the research that we're doing and the findings that we've made. So, being that I'm not a clinician, it's always good for me to remind myself why I'm doing what I'm doing. I'm a PhD chemist in a translational research subject, and so I'm trying to take the approaches for collecting large data sets in an unbiased fashion and applying it to a pragmatic topic, in this case, understanding diabetes, either factors that would portend progression of the disease or development of the disease, or possibly a development of complications of the disease. So in the upper left-hand corner, there's 3 arrows that are pointing up, and below each one of those vertical arrows is an abbreviation. Is A1C, FPG, and OGTT. So these are 3 different metrics that could be used clinically to identify whether or not somebody has diabetes. So the first being hemoglobin A1C, or kind of a steady state quantification for the extent of glycosylation of hemoglobin. So this is kind of gives you a rolling average over some extended period of time for the glucose levels in the blood. The second value is the fasting plasma glucose level. This is kind of a measure of how well your body is doing to handle glucose and pull it into your major target organs, muscle or liver, to form glycogen when it's sensed to be too high. And the last is the oral glucose tolerance test, which is kind of a timed assay where a bolus of glucose is delivered, and then usually 2 hours after that delivery, the levels of glucose is assessed in the blood. So for all of these things, we have really defined metrics for identifying the individuals for diabetes. And later on, my talk kind of goes towards saying, although we can identify diabetes, we cannot easily identify the complications of diabetes. And so that's why we need these other methods. So you'll notice from the lower left-hand panel, the number of deaths for leading causes of death. Heart disease is number 1, and about 5, 6 below that is diabetes. But we all know that diabetes is associated with or related to heart disease. So a lot of these are all interrelated numbers, but what that shows us is that if within the United States annually, there are a large number of deaths associated with diabetes and complications. The right-hand side panel is just a projection in 2045, the rate of increase of diabetes. And so it is a problem, and it's going to remain a problem. So within the realm of kidney disease, so my faculty appointment is in nephrology and hypertension, so I try to always bring it back to that target organ to make sure that the other clinicians in that department know that I'm actually doing something worthwhile, not playing around. So here you see about some data published in 2006 for type 2 diabetics, all with microbiome. These are 7 different studies encompassing individuals in the United States and Europe, around the world, looking at people that have a EGFR, an estimated glomerular filtration rate of less than 60 mils per minute, which would be people that are CKD stage 3 or lower. And you'll see below each one of the studies the number of individuals within that study. So we go from hundreds to thousands, tens of thousands. The percentages of the individuals that had CKD stage 3 or lower, and then individuals with some measure of albuminuria. And then below each one of those kind of Venn diagrams, you'll see a listing of the percentage of individuals with no CKD. So what the take-home message for that, for me, is that no matter where you are on the globe, individuals with diabetes, a large fraction of them do have chronic kidney disease. So that's something that we have to deal with. And chronic kidney disease is kind of a difficult subject to happen upon, because usually people are diagnosed with that when they present to their primary care physician, provide a urine sample, and they just happen to check for protein or serum creatinine from their blood samples. So it's kind of a silent disease in that regard. So how does diabetes impact the kidney? How does diabetic kidney disease develop? Classically, the major boogeyman has always been reactive oxygen species. So the idea being that the kidney will freely ultrafilter glucose, and when you have poorly controlled glucose levels in your blood, they can hit in an average person 25 millimolars. And so what comes through the filtration device barrier that bathes the podocytes and the tubular epithelial cells and the sanguinal cells in a ultrafiltrate containing up to 25 millimolar glucose. Of course, in those people that have really wild swings, that value can go much, much higher than that. In an average individual with well-controlled blood glucose levels, those blood glucose levels are usually around 5 millimolar. And so whenever we do experiments with cells in culture, renal cells in culture, we usually think of 5 millimolar glucose in the culture media as being somewhat normal and 25 millimolar as being high. So the cells freely take up glucose, and in fact, the proximal tubular epithelial cells and the distal both have a sodium glucose cotransporter that is scavenging glucose and sodium pretty voraciously. And so all of that glucose gets pulled in and starts to get metabolized through a variety of pathways. Here you can see the 4 major pathways that people tend to talk about with regard to toxicity to the renal parenchyma. The polyol pathway, where glucose is reduced into sorbitol and then reduced a second time into fructose. There's the hexosamine pathway, where glucosamine 6-phosphate is formed and converted into UDP glycnac. There's the PKC, protein kinase C pathway that leads to the formation of glycerol phosphate, diacylglycerol, and activation of protein kinase C, which can activate the NF-kappa B pathway. And then there's the advanced glycation end product pathway with the formation of methylglyoxal, the modification of proteins to form the advanced glycation end product, modified proteins that bind to the receptors. And so that all activates generation of reactive oxygen species, generates signals for apoptosis, that generates signals for secretion of chemokines, cytokines that drive fibrotic pathways. There is distinct glomerular involvement in this disease. This is a cross-section for a glomerulus, the filtration apparatus for the kidney. We have the afferent arteriole, the capillary bed, and then the efferent arteriole, so blood coming out, blood going out, coming in, going out. Along this filtration barrier, we have the endothelial cells, we have the fenestrated basement membrane, we have the podocytes, and between the podocytes are the foot processes where the slit diaphragms are found. And so anything with a molecular weight of 20,000 or less is freely filtered, and that flows down the nephron through the tubular epithelium. If we have damage to this filtration barrier, then the podocyte foot processes are lost, the podocytes become activated, they can release into the urine, and so this is a podocytopathy, and we have then filtration of albumin into the urine space, large amounts of albumin. Normally the proximal tubule will pick up that free protein. There are the megalin and cubulin protein scavenging receptors, but they can become saturated and eventually you lose the capacity to scavenge that protein. So the combination of glucose levels and protein being ultra-filtered can drive activation of the mesangial cells, loss of the podocytes, and in the tubular epithelium, recruitment of activated fibroblasts that drive tubular interstitial fibrosis. The fibrosis of the glomerular tuft and fibrosis of the tubular interstitium are hallmarks for diabetic kidney disease. And so classically, that's what you had, was glucose going to ROS, or protein toxicity, activation of a few cell types, and fibrosis. So I have to ask myself, given that we thoroughly understand everything about this, why do we want to expend any effort to study diabetic kidney disease? And I want to give you 2 examples of what motivates me to spend time thinking about this. So this is an example of a histopathologic section of a kidney biopsy illustrating a healthy glomerulus. So this is a human biopsy. It's obviously been optically enlarged, but you can see the lacy capillary loops through here. You see nuclei stained in blue, collagen stained in the pinkish color, and in the diabetic, the biopsy of the individual with diabetic nephropathy, you can notice one thing is that there's been some hypertrophy of the glomerulus. It's large, and you'll notice that there are these pinkish nodules in here, these Chemostone-Wilson nodules, and you can see a lot more pink in the middle, so we have fibrosis. And if you actually did electron microscopy and looked at the width of that basement membrane, you would see in diabetes it's dramatically enhanced. And so, mostly people consider this to be a terminal process. Once it's occurred, it's not going to be reversed. But Paula Fioretto and Mike Maurer have a protocol to surveil individuals receiving pancreas transplants subsequent to the transplant process. And so here are 3 examples of biopsies from the same individual, biopsies of their kidneys after they had received a pancreas transplant, or actually column A is before. And what you can see is that before receiving the pancreas transplant, the H&E stained tissue section for the glomerulus shows the Chemostone-Wilson nodules and shows the areas of fibrosis in this glomerular cross-section. And there's also some areas within here of showing tubular damage, mainly brush border loss. You don't have the fluffiness to the brush border epithelial, proximal tubular epithelial cells and their brush borders. So 5 years after pancreas transplant in this individual, you'll notice that there is not a large change in the amount of fibrosis and possibly a reduction in the Chemostone-Wilson nodule appearance, but it was not significant at all. Then also between these 2 biopsies, there was no change in the glomerular basement thickness, the total mesangium, which is this pink area in here. There was no change in the mesangial to glomerular volume ratios, the mesangial cells that are in here that regulate capillary surface area through contraction or relaxation. There was no change in the tubular basement membrane thickness, and there was actually an increase in the interstitial space in between the tubules. But after 10 years in the same individual, there was a significant decrease in all of those values. So pancreas transplant and reestablishment of a normal glucose level without the need of exogenous glucose did in fact reverse the disease, and the findings at 5 years were attributed mainly to the anti-rejection drugs that the individual has received. So this to me is an indication that if we understand what's going on, there might be an opportunity to reverse the process in an individual, not in a mouse, not in cell culture. The other is the Diabetes Control and Complications Trial, the DCCT, and I've thrown this up here last year, so I won't go through it much more. Dr. Mokshagundam is much more of an expert on this, and he has participated in this, so if you have questions, ask him. But this was really the first clinical trial to address whether or not tight control of glucose levels actually had a benefit over just your standard approaches to controlling your blood glucose levels. So this started in 1978 with an RFA for request for proposals and funding. The development of the consortium occurred between 83 and 89, where they had a time for planning of this clinical trial, establishing the feasibility of what they were doing. They had a recruitment period, and then they had the actual trial period. After the trial period, all of the individuals that were recruited within the study, some of them agreed to participate in a second follow-up trial, the Epidemiology of Diabetes Interventions and Complications, or the Edict Trial, which went on for about 27 years. So in this study, in the DCCT study, there was a randomization of individuals that met recruitment characteristics to one arm that received conventional therapy and another arm that received intensive therapy. And intensive therapy was individuals seeing their endocrinologist much more frequently, measuring blood glucose levels much more frequently, giving themselves or being given insulin to more tightly control blood glucose levels. And what was observed was immediately that there was an immediate benefit. People hit their targets of hemoglobin A1C to 7% or thereabouts, and a significant reduction from what they started at. And at the end of the DCCT trial period, the people that had been under intensive control were released from that intensive control. And it was mainly if they maintained that themselves, then they maintained it. There was no way to monitor that. But what was observed was for a period of about 3 to 4 years, the metrics of hemoglobin A1C were maintained across that population to be significantly lower, even though presumably they weren't doing anything different. And so this brought in the concept of metabolic memory. In other words, during these 10 years of intensive treatment, your body had programmed, maybe the genetic response had been trained to have a complement of gene expression programs that were beneficial toward minimizing complications. And so long-term impact, whether you're talking about retinopathy, nephropathy, cardiovascular outcomes, mortality outcomes, blood pressure control, these were all reduced as a function of intensive glucose control. So those are the two things that give me hope that we can actually, if we learn something really cool that's meaningful, we might have an impact. Clinicians, you have impacts every day. Researchers, impacting patients. It's pretty rare, so we enjoy it when we get that chance. So central dogma of biology, I want to transition over into the concept of epigenetics and epi transcriptomics. And so the central dogma of biology is kind of taken to be gospel, right? So it's like the sun rises in the east and sets in the west, right? So the central dogma of biology is you have DNA. DNA can replicate itself. DNA can be transcribed into RNA, and RNA is translated into protein. And that's as simple as it is. But we know dogma evolves. And these days, there is this concept of epigenetics. And the significance of epigenetics is that it's a dynamic process where you have modifications to DNA or modifications to RNA or proteins that are reversible. And so the thought for metabolic memory is that you have somehow programmed gene expression programs as a function of epigenetics to provide that supporting role for minimizing diabetic complications. So this is just an example of a chromosome packed down into these histone-rich cores, nucleosomes with the DNA wrapped around it, and then those wrapped around themselves again to form kind of this compact structure that allows the DNA to be packaged into cells. But in effect, what you also have is quaternary structure, tertiary structure, secondary structure. You have a variety of levels of structural biology here that can contribute to transcription. So in this little box here, you see the DNA being wound around the histone cores. And so that DNA is where you can have methylation take place. The proteins here, the histone tails, can be methylated, acetylated, phosphorylated, ubiquitinated, etc. And then the RNA itself, once it's made, it can be post-translationally modified, post-transcriptionally modified, and that modification can influence translation rates or degradation rates. So epigenetics, it used to be that this, around the year 2000, 2005, this little image was all over the place because the concept is you have a caterpillar, you have pupae larvae, you have the mature butterfly. the same organism, same genome, different representation of the proteome. So you can have conditions in development or under other circumstances where you have the same genome, it's just the expression of that genome is altered by the environment. So practical examples of epigenetics impacting your health. Example, an infection with tuberculosis, how histone modification can impact an interleukins expression. And colorectal cancer, there's actually an increased level of DNA methylation of CEP9. And nutrition during pregnancy, during World War II, in the Netherlands, there was a winter famine, and individuals that were born during that time were followed throughout their lifetime, and it was noted an increased prevalence of heart disease, schizophrenia, and type 2 diabetes in these individuals that were associated with DNA methylation as well. Epigenetics can change in development, in age, and in habits. Your own personal habits can impact, epigenetically impact your health. So for diabetic kidney disease, we have the classical inputs of metabolism and hemodynamics. So with hemodynamics, we have mechanical shear stress, or we may have humoral generation of angiotensins. Metabolically, we have hyperglycemia, which impacts cytokines, ages. We have dyslipidemia, so we have oxidized LDL or non-esterified fatty acids binding to their cognate receptors. And our classical outputs that traditionally we studied were inflammation, proliferation, differentiation, hypertrophy, apoptosis, all these things. But actually, we do have an input here for epigenetics. And that's whether it's epigenetics that's addressing modifications to those components that I've already described, or actual changes to transcription factors and how they interact with chromatin. We have microRNA, so we have non-coding RNA generation that's impacting gene expression. So anything that would affect the modifications, relative or spatial abundance of transcription factors, or expression or abundance of non-coding RNAs, those can fall under the umbrella of epigenetics. So here's a nice little complex and confusing diagram for molecular hallmarks of epigenetic control. Around the purple central wheel, we have kind of a topical identifier for the molecular hallmark. So enzymes that control the writing or erasing of marks on the histone. We have cofactors that are associated with providing those modifications. So, for instance, S-adenosylmethionine, providing the methyl group for methylation, etc. We have metabolites that are associated with substrates in the metabolic pathways associated with cancer and inflammation. The histones themselves, in some cases, there are pathogens that have histone mimetics that can mimic histones. Non-coding RNAs, remodeling, DNA methylation, and a variety of conditions that are associated with these marks. But we want to layer in here the Epi Transcriptome. And so this is this image that popped up is not meant to be read. It's meant to give you some measure of what we're talking about specifically when we talk about Epi Transcriptome. So this illustration has 4 regions on the outside: the yellow, the purple, the blue, and the other purple, mauve, whatever. These are starting from the upper left-hand corner: adenine, guanine, cytosine, and uracil derivatives. So these are all nucleosides. So these are the nitrogen bases plus the ribose. And you'll notice that there are, in each one of these structures, there's a red component and a black component. The black component would be the unmodified version The red would be a modification that's been introduced as a result of enzymatic or non-enzymatic activity. So when we're wanting to study the Epi Transcriptome, we can either, right now, most of our ability to do this is at the level of individual bases. As you know, if you're trying to sequence a gene, you actually need the linear combination of bases to understand what the gene is. So right now we have direct methods for understanding the bulk prevalence of modification to individual bases. And people such as Dr. Zhang are developing methods that would allow them to have short but consecutive regions of bases to understand the organization of the modifications. So the transcriptome, the set of all RNA transcripts in a cell, including coding and non-coding RNA strands. The Epi Transcriptome would address the chemical modifications that modulate the effects. So I want to transition into the last part of the talk where I discuss the work of Zoe Hack over this summer. This work would not have been possible without collaborations. Dr. Hetman in environmental medicine, Dr. Kling, Dr. Zhang on main campus, as well as colleagues in my group, Madhvi Rain and Michelle Barati. So first, there's basically three parts to what I want to show you. This is all pilot study. This is all pilot study in examining a model of type 2 diabetic kidney disease. The first thing we did was a targeted study using an immunoblotting approach to determine if specific enzymes that either write in the modification, erase the modification, or read the modification were affected in our model. And based upon those results, we were encouraged to go ahead and do a discovery study to kind of ascertain globally what are all the modifications present. And then based upon those two results, we went ahead and did a discovery proteomic study. And I call these a discovery proteomic study because it was not as in-depth of a proteomic study as we could have done, meaning that it was a very short LC-MS gradient time on the mass spectrometer. So the model that we're studying is the Dbdb model of type 2 diabetes. And so Db stands for diabetic gene. This is a gene that, this is a model that results from a G to a T point mutation of the leptin receptor in adipocytes. And this results in an abnormal splicing and a defective signaling of the hormone leptin. So these animals that are homozygous for this mutation have hyperphagia. So they eat and they eat and they eat until they exhaust the insulin signaling and they become obese. So this was studied as far as diabetic kidney disease, 20 years ago by Kumar Sharma. And so table 1 and table 2 present some parameters that I'll go through, and figures A and B illustrate on the left a heterozygous animal illustrating a normal glomerular cross-section, and a homozygous individual animal that illustrates diabetic kidney disease, all the fibrosis, the mesangial sclerosis that's present. So on table 1, the time point that I want to point out is age week 12, because Dr. Hellman provided us kidneys from animals age week 12 to 14. So at 12 weeks of age, the glomerular surface area of the Dbdb homozygous versus the DBM heterozygous is significantly different. The glomerular surface area is increased. So that's portends diabetic kidney disease. The mesangial matrix area was also significantly increased. And when we look at the amount of urinary output of albumin between the homozygous and the heterozygous, it's incredibly significant. So at 12 weeks, the animals are diabetic and they do have diabetic kidney disease. So Dr. Hellman studies the effect of exercise on the cardiovascular system. So when I contacted him and said, I really want to study Epi Transcriptomics, Dr. Zhang and Li Qinghe said, we need lots of RNA. And I said, crap. So I need a whole kidney. I'm not an animal person. I'm a kidney person. I respect all of you animal people. He had saved kidneys from his study, which is kind of the responsible thing to do. If you're studying one system and you sacrifice a mouse, a lot of people will collaborate and farm out all those other organs. So he provided me with 4 kidneys from each one of these 4 groups of animals. So we have the heterozygous that lived a sedentary life. We have a heterozygous that at 12 weeks of age started being exercised 5 days a week by being placed on a treadmill. The first week it was 40 minutes a day. The second week was 50 minutes a day. The third and fourth week was 60 minutes a day. That was Monday through Friday. They had the weekends off. So I was intrigued by this, and we know from diabetes that exercise is an intervention. You know, diabetes is get up, get moving. How many minutes a week is it? 120, 150 minutes a week is what you need.
Diabetes.
Yep. So
Type 2 diabetes.
okay. Okay, so that time is
Diabetes prevention program.
DBDb.
1K with nerves. That has increased younger individuals.
really
And type 2 is
okay.
cardio and
So this would be, I don't know what treadmill is, cardio or endurance or both.
start on the weekend.
So in the kidney field, exercise is encouraged. This is the National Kidney Foundation. They say, yeah, get out there and do it. But when you look in the literature, there's nothing to say it improves proteinuria or eGFR. In fact, some people kind of feel weird about it because a lot of people that do extreme exercises, they either take creatinine, so they're going to get a bump on their serum creatinine, which is going to look like their kidney function went down, or there are people that do these endurance things and they actually damage their kidneys, so they have a little bit of hemoglobin spillover, a little bit of blood spillover. So there's actually no clear understanding of does exercise positively benefit the kidney. And I don't think there have been any clinical trials in it because I don't think there's any money in it. The one thing that is understood is that if you're CKD stage 5, ESRD, that exercise, regular exercise, weekly exercise does improve your quality of life. So in people on dialysis, that's a major thing. That's a major thing to improve their quality of life. So what Zoeb did was to take the mice kidneys, work with Dr. Barati. Michelle is kind of she's our director for microscopy, so she does all the histology, all the confocal microscopy type stuff. And so we need that capacity to validate any of our findings. She also worked through the RNA extraction and protein isolation with Zoeb. And the RNA, we went through and assessed the integrity of the RNA using the bioanalyzer. And what we found was that out of our 16 samples, 2 of the samples that originated from the mice that were diabetic and had exercise, there was some degradation of that RNA. And that impacted us because the original hypothesis, which I'll show you some background data for, was to focus on the most prevalent modification, M6A. And we wanted to basically learn from Dr. Kling and her success and do a targeted analysis of messenger RNA. But with this result, I knew that we could not do that because we had some degradation. But we did a global analysis. And in the global analysis, what you basically do is you use a enzyme that's going to cut up all of the RNA into individual nucleotides, and then you use an enzyme that removes the phosphate, so you have individual nucleosides. So the fact that these were already partially degraded bothered me none at all, because we were going to completely degrade everything. So that was submitted for analysis at CREAM. Meanwhile, Zohaib worked with Dr. Rain to learn immunoblotting techniques, and we focused on 4 proteins. So what we learned is that diabetes and exercise, how they affect the RNAome, is we know that there is literature to support the fact that M6A levels are significantly increased in diabetic kidney biopsies compared to healthy biopsies. Right? So there was somebody that had that data out there. So we knew that M6A or Epi Transcriptomic marks were impacted by diabetes in people. These are mice, so there's no guarantee, but that's supporting data. We also know that the enzyme that's the catalytic component of the core proteins that introduce the M6A was actually found to be altered as a function of exercise, but this was in brain and in muscle cells. So we're studying kidneys, so there's no guarantee. So M6A, what does it do? Just a real quick cap on this. It's a modification to adenine, and it contributes to the regulation of RNA expression, splicing, transport, stability, and various processes that include circadian rhythm, energy homeostasis, obesity, and cancer. There are 3 big groups of enzymes: the writers, which introduce that methyl group onto that number 6 amino group, the erasers that remove it, and the readers that somehow interact with it and are directed towards some functional change in their chemistry. So this is a layout of all the readers, writers, and erasers that are involved with M6A biology. So this is, everything in red is a writer, everything in green is an eraser, everything in blue is a reader. And so based upon our readings and based upon the amount of money that I had to buy antibodies, we selected metal-3 as a target for our target analysis. We identified FTO as a target for our eraser, and we identified insulin growth factor 2 binding protein 2 as a target for our eraser study, as well as HN RNPA 2B1. And I've come to not like these class of proteins because I don't remember or understand what it means. So jumping right into the data, so we have 12 samples that were analyzed, 12 animals, so 4 groups of 3. And we chose to do it this way because we could fit all of those samples plus a molecular weight standard on one gel for one blot. And I didn't have to try to worry about putting those other 4 onto a second blot and somehow normalize the data and integrate it, right? So red is data for the metal-3 normalized to the actin, beta-actin in that lane. The black is for the heterozygous, normal, wild type, whatever you want to call it, metal-3 normalized actin. I ran this as a 2-way ANOVA analysis, but if a pure statistician is in here, please ignore that I did this, because what I hear is that you need at least 9 to 10 per group to actually do a robust 2-way ANOVA analysis. So anytime, I'm assuming that there's one person here who doesn't know what I'm talking about. So if you have 2 groups and you want to do a statistical analysis, you do a t-test, right? And all you're checking to see is if the differences that you observe are randomly different or if that's a true difference. So if you have 3 groups or more, you have to do an analysis of variance. And if you have a 2x2 or style of design, then you may want to see if one condition interacts with the other condition such that the interaction makes something worse or makes something better. And so that's the interaction term here. So the sedentary animals, the diabetic animals had more of metal 3 than the non-diabetics. And they had this in both the sedentary and the exercise. So the effect of diabetes was statistically significant. So this statistical significance includes the difference here as well as the difference here. The effect of exercise was not statistically significant. The interaction term between these two was statistically significant. The difference here and the difference here was considered to be significant. So exercise and diabetes both affect metal 3 expression, and they do it in the opposite way, and that's in fact consistent with what the literature said should happen. Nobody studied the two together, but each independently, this would have fit with that perfectly. Our antibody showed a second, so these are two different blots here. This isn't one blot. So this is a blot, and the lower tracing is a blot. So here, the antibody recognized two bands. The 65-ish, 67-ish is where metal 3 should be. There was another band of around 150. I don't know if anybody else in here has ever seen that with their antibodies, but I really don't have an explanation for what that one is. So this other reader, HNRNPA2B1, there was no statistically significant differences here. We did note that two of the animals that were in the diabetic intervention group, the bands were obviously different than the others, which might suggest some sort of degradation of those proteins. I have no idea what that means. Haven't thought about it. So FTO stands for iron 2 and alpha ketoglutarate dependent fat mass and obesity associated protein, FTO. It is a protein that after metal 3 puts on the methyl group, FTO can oxidize it and remove it. So it's an eraser. We did not see anything significantly different here. There were two bands. This should be the expected band. I did the statistics for both bands. There was nothing significant. Lastly, the other reader is IGF-2, BP2, insulin growth factor 2, binding protein 2. And we see that the sedentary animals, the heterozygote, had much more than the diabetic animal. We see that exercise, there is a significant difference between these two and these two together. So exercise had a significant difference. Diabetes had a significant effect, and there was an interaction term. So IGF-2, BP2 is also known as IMP2, and it binds to and inhibits the translation of a large number of messenger RNAs. It just so happens that the first one that was studied was insulin growth factor 2, IGF-2. Since then, there have been a variety of others. So IMP2 loss mitigates. So somebody looked at, in a knockout animal, the effect of IMP2 loss, and they observed the mitigation of the development of obesity, fatty liver, glucose intolerance, et cetera, in those animals. And so that might be consistent with the exercise arm of this. It did have an effect on the expression of uncoupling protein 1 and on adipose tissue as well. So this is actually something that's an interesting protein. It's genomically imprinted, which means to say the copy of your IGF-2, BP2 gene from one of your parents is expressed and the other one is not expressed. And it's always the male copy, the parental copy that's expressed. So that's our data on our targeted analysis. And we saw half of our readers and writers were significantly altered by diabetes and exercise, thus suggesting that we should see globally changes. And so here we're looking at global changes, and you would expect that most of the modifications that we're going to see are going to come from ribosomal RNA, because there's more of that as a mass fraction, and then transfer RNA, and then messenger RNA. And it turns out transfer RNA has a lot of modifications, So we saw 38 observed modifications across those 16 samples. So we analyzed all 16 samples here. So they conducted a series of pairwise comparisons to ascertain whether or not there was a significant change. And we filtered this based upon a p-value of, that should be p less than 0.05, not p greater than 0.05, and a fold change filter. So these are the 5 conditions that I consider to be important. Looking at the effect of exercise on the non-diabetic and the effect of exercise on the diabetic. Looking at the effect of diabetes on the sedentary. Look at the effect of diabetes on the exercise. And then kind of, another combination where both things change. So what we did was to filter everything, all the comparisons by p-value of less than 0.05, so that got rid of a lot of things. Then we filtered by a fold change, log2 fold change of smaller than 0.25 or greater than positive 0.25. And we saw that there were 22 different significant changes. So this is a lot of data. Usually when you have this type of data, you just look for the biggest changes, because those should be the most repeatable and the biggest effect. And so this 2-thiouracil is a big one to study. It's involved in tRNA modification and base pair wobble. So there's a way to introduce mutations within the translation of your gene by having tRNA, amino acyl tRNA, bind to the binding pocket inappropriately because there's wobble there. The first 2 bases in the codon bind tightly, and the third one has a weak binding. So the ones that we're actually very much interested in are in the diabetic animals, and so I'm going to point out all the things that are in the darker colors. So we saw in the exercise animal, there was more of this hydroxymethyl-6-adenosine. So this is a M6A derivative, and based upon our immunoblot data, we might think that there should be more M6A, but we didn't see more M6A, but we saw more of its products of degradation or further modification. DHU is dihydroxyuracil. This is an oxidative damage byproduct. The fact that this has a negative sign means that the numerator was smaller and the denominator was bigger. So this would be in the diabetic animals that sat around. They had more oxidative damage, possibly. Here we have MS2T6A. This is another modification of adenosine. These things that have lots of abbreviations and numbers are considered to be heavily modified and do introduce usually more wobble into the tRNA. That's about all I can say about it specifically. This is a modification that is observed in transfer RNA for lysine. So it's a modification introduced by this protein CDKAL1. People have studied CDKL1 knockout animals, and they have observed that in the knockout animal, there is pancreatic islet hypertrophy, there's decreased proinsulin synthesis, decreased insulin secretion, and impaired blood glucose control. So if there is more of this in the exercise, I would assume that there would be more insulin control. And lastly, we have more cytosine. And this one is kind of hard to impute what the meaning of it is because this is one of the 4 major bases. But there was an increase in the amount of this on a total level in individuals with exercise. So cytosine is a substrate for cytidine deaminase that converts it to uracil. So uracil is made from C. And deficiency of this enzyme would result in more of this. And deficiency accelerates autoimmune diabetes in the NOD mouse. I don't know how to interpret that here, because there's more of this in the exercise animal. Because this does funnel out to uracil and other cytidine derivatives, it may mean that there's flux, metabolic flux going other places. Right? So regulation of gene expression by RNA modifications. This is to recap what I've been trying to say, try to make it visual. You can have modifications that are introduced by writers, modifications that are removed by erasers. That modification can be bound by proteins and stabilize it, bound by other proteins and promote decay of it, or you can scaffold across and initiate translation. So these modifications are multifaceted. And one of the most intriguing ones to me is actually because we have global data and most of our data are modifications observed in transfer RNA. These modifications can affect translational rate, biogenesis, codon optimization, that's the Wobble, and biogenesis. Right, so we address these hypotheses that regulation of expression could impact the renal proteome. I'll go through this pretty fast because I talk slow and we're losing time. We collected 16 data sets. We compared those 16 proteomic data sets. We identified about 1,500 proteins. About 11 of them were identified consistently enough to allow for comparison. This is a principal components type analysis plot. The data points that are in red are from the diabetic animals. The data points that are in blue are from the heterozygote normal animals or non-diabetic animals. When we compare the diabetes, not considering exercise, just the Dbdb versus DBM, we see three groups of animals. So these two are actually mainly the diabetics, the wild type with exercise and the wild type without exercise. So those are coming through that exercise has an effect on the proteome in the wild type that's observable independently, whereas in the diabetics it really doesn't. When we actually do our comparison to compare exercise versus sedentary, we actually see all of our groups tease out on this level. So when we do our statistical analysis of this, we're definitely going to have a primary comparison to look at exercise, the effect of exercise. So this is just a volcano plot of diabetic versus non-diabetic animals. So this would somewhat ignore the effect of exercise. So you can see from the volcano plot, two groups of 8, we've got a large number of things that are upregulated in diabetes and a large number of things that are downregulated in diabetes. So one of the things I did was to look at this from protein-protein interaction analysis, filtering out proteins for mitochondrial, peroxisomal, and proteasomal enzymes to try to deal with what's known about transfer RNA regulated in diabetes, mainly affecting mitochondrial gene expression. A recent publication of individuals that compared the proteomes of DB and DBM animals for the kidney identified large changes in the peroxisome, and then the proteasome is one route for degrading proteins. And what we can see is the differential abundance in the diabetic animals is color-coded by this halo of blue and red. So a blue halo indicates increased in the diabetic animals. The red halo is decreased in the diabetic animals. The red bubbles are for mitochondria, the blue bubbles are for peroxisome, and the greens are for proteasome. And what we can see is that the proteasomal components that are regulated in our animals are decreased in the diabetics. The mitochondrial proteins that are regulated in our animals are increased in the DB animals, and the peroxisomals are largely decreased. So I looked at the differentially abundant proteins based upon relative abundance and segmented them out based upon their direction of the expression in each one of the 4 groups. The DBM with sedentary, the DBM with exercise, the Dbdb with sedentary and exercise, whether things went increased in that condition or decreased in that condition. And this is a representation of differentially abundant proteins out of those 1,100. So I looked at the gene symbols for the proteins that were regulated in each condition, and I highlighted in black those proteins that are known to be RNA interacting proteins, because that's what I really wanted to know was if we're looking at epitranscriptomic effects and we want to know how these conditions are affecting the epitranscriptome, we need to monitor RNA interacting proteins. And what you can see is that exercise in the heterozygote, we had a large fraction of those that were regulated are RNA interacting proteins. And in the Dbdb exercise, the proteins that went down, that decreased, a large number of those are RNA interacting proteins. So I think going forward, we're going to develop a hypothesis for this on how these are contributing to the development of kidney disease, if at all. The last thing I did was to look at all of those regulated proteins and assess what could we impute based upon their identity for likely microRNA and likely transcription factors that are driving the changes. One of the things I can say is that if you look down here in those proteins that are increased in diabetes, the number one protein transcription factor for driving that expression is SREBP1, which we would expect. So that gives me confidence in this data. I don't know enough about miRNA and haven't had time to dig into all of these to know whether or not there's supporting data to say that we believe it or not. So a summary of results: diabetes and exercise are associated with altered levels of M6A readers and writers. They both affect and impact the global nucleoside modification, likely impacting tRNA and mitochondrial proteome abundance. And we have some data to impute likely non-coding RNA and transcription factors that are involved in the mechanism. Future directions are going to try to elucidate one of these three aspects of how the external stimuli of diabetes or exercise is contributing to the epitranscriptomic regulation of diabetic kidney disease. Perhaps metabolic memory is associated with epitranscriptomic alterations, and blah, blah, blah. A lot of people to thank. Zohaib did a lot on this project. Danny and Ming helped. Sabine helped with the bioanalyzer. Could not have done this without all of these collaborators and the funding. So I'll be happy to take any questions you may have. Here's your today's CME code. Please don't forget to get your credit. Thank you. Yes, ma'am.
Wow, what an interesting story. You've really done a lot. I'm really proud of it. It really seems like tRNAs are very interesting.
Yes.
Anybody who's a tRNA, I guess I should go look at them and find somebody to bring this in as this data that suggests there's interesting.
Yeah.
I thought it was very nice that you found some similar, but some different changes in that transcript. But and I don't know what to do with those bases either. I just ignored them because I thought, well, you're digesting everything, and the bases are different. Well, I don't know what that means, so I just shut them out of the data.
Yeah, so the whole thing with an omics approach is you're building a hypothesis. I think Dr. Kling was making a comment that we had some data that was consistent internally within itself and consistent with things that she's seen, but the whole field of transfer RNA is completely new or new within the past 10 or 20 years, and it might be pertinent to get somebody in to speak on that. And then there's an aspect of having a large number of modified bases that are really an unknown on what to do with or how to handle because their metabolism hasn't been fleshed out. So there are some comments. You have a question?
No, there are several questions.
Yep. So this is from Girish Kotwald. I was intrigued by the data showing pancreatic transplant could potentially reverse the structure of the kidney, but was there a significant reduction in proteinuria and GFR? Yes, there was a significant improvement in GFR. I don't remember what level of proteinuria they had. I'm not sure if they would have received a single, just a pancreas transplant if they had had any amount of proteinuria. Usually they will do a double transplant at the same time if there's any degree of proteinuria. So another question, are there miRNA differences noted in diabetic kidneys and normal kidneys? Absolutely. Rama Natarajan at Duarte City of Hope is an expert in this area. And lastly, does the microbiome gut flora influence kidney disease? This is a hot topic within the past 12 to 18 months, and there are some there's a really nice review in Nature Reviews Nephrology that addresses this. So as far as I can see, those are the three questions that are online. Yes, ma'am.
It's interesting. The question is, you eliminate mitochondria, mitochondria and Ross blank. Lot to look at.
Oh, no, I meant to say that, so Dr. Clark was asking about the angle of mitochondrial alterations. And what I wanted to say, I apologize if I didn't say it, is I think mitochondria is a significant component in all the data that we have here. That there's a specific subset of tRNA modifications that are incredibly important for the translation of mitochondrial genes. And we're seeing regulation of nucleosides that would be specifically involved in mitochondrial genome expression, and we're seeing regulation of mitochondrial proteome. So yes.
I thought you said that in proteome you were.
Yeah, I apologize. I was rushing at the end because I'm long. I'm over time as usual. So, please.
It was very interesting.
Apologies. The p-values that we use for significance are always 0.05 or lower.
Yeah.
The statistics that were sprinkled about with the immunoblots, we did the two-way ANOVA, and for anything to be significant, it had to be less than 0.05.
I just thought you said maybe 0.25.
That was probably a mumble again.
I think that's a log-fold change.
Oh, log two-fold change. Yes, log two-fold change was, so when we take the ratio of abundance, the net ratio, we take the logarithm 2 of that.
I'm going.
The reason we do that is if we want to put that on a graph, it's hard to graph numbers above 1 and numbers that are just 0.0 something. So when you convert it to a log 2, when the numerator is larger, that value is positive. When the denominator is larger, the value is negative, and they normalize each other out. So if there's a double this way, the half will have the same numerical value. So that was 0.25.
Okay.
Yeah. And some people use a much larger value for significance on that, but that's usually because they have much bigger changes. And we're looking at a global change in nucleosides. And based upon the instrument that was used and the sensitivity, we're only going to be observing the dominant species. And so you're not going to see tenfold changes. Yeah. Oh,
But IGF-BP, all the things that you studied that was binding protein.
so IGF-2 BP2 has, from GWAS studies in diabetics, there are SNPs that are associated with diabetes. It's not well understood itself. It binds to the 5' end of IGF-2, but it also binds to, now that we know, a large number of other transcripts. And so how that impacts here, I don't know. I know IGF-2 has a role in diabetic complications as well. So it's interesting that it goes, that the direction of change with
I thought it would be the other way.
yeah, so in the diabetic animal versus the non-diabetic animal in the sedentary, the expression isn't consistent with the literature. But with exercise, there's a direct change in, yeah. So I don't know what that means.
Yeah, I'll have to go back.
I don't know if there's something with, and the other thing that I need to work with Dr. Hellman on is the one protein. If you look at the diabetic animals that went on exercise regimen, there were 3 genes, 3 proteins that were increased, that were not increased in the others, but were increased in those. And the highest one was hexokinase 1. And that is the enzyme that phosphorylates glucose to shove it in glycolysis. And why exercise in the diabetic went up when it wasn't in the wild type? I would have, maybe it's just a combination of multiple factors. I don't know. So there are some intriguing things in the data set.
That's right.
So, thank you. Yeah. Well, yeah, if Dr. Winters is still on, I hope he has a safe place to find down there. I think that.
Thanks, Mike. I happen to be in.
Category 4 and 5.
I happen to be in Pittsburgh.
Very bad. He's where?
He's in Pittsburgh.
He's in Pittsburgh. Yeah, he's safe there. The only thing worse than that category 4 is the stop. Okay. Thank you very much.